Chapter 1. A Cell and the Idea of a Day. Before we follow what a human cell does over the course of a day, it helps to slow down and ask a strange question. What does a day mean to a cell? Because a cell does not experience time in the way we do. For us, a day has edges. We wake, we eat, we move through rooms, we feel morning arrive and evening settle, we get tired, and eventually we sleep. Cell has none of that. Inside a cell there is no sunrise, no darkness, no silence, no switch that flips. There is only continuous chemistry, and it happens in a space that is almost unimaginably small. A typical human cell is only a few tens of micrometers across, something like 1/100th the width of a human hair. Yet inside that tiny space, there is almost no empty room at all. It is packed. If you could shrink yourself down and step inside, it would not feel like floating in open water. It would feel like moving through a warm, crowded sea. Water fills most of the volume, but it is not simple water. It is water thick with dissolved salts, sugars, metabolites, and proteins. Every direction is occupied. Large molecules drift slowly, bumping and sliding past one another, not because the cell is calm, but because it is full. This crowding changes what movement means. At our scale, a molecule seems like a point. At the cell scale, molecules are objects with shape. They collide. They stick briefly. They separate. The inside of the cell is not a clean laboratory. It is a dense mixture where reactions happen because the laws of chemistry make them likely. There is no awareness here, no planning, no intention, just probability and constraint. And yet, out of this crowded motion, stability emerges. A living cell maintains its internal conditions within narrow limits. Acidity, ion concentrations, the balance of water, the availability of usable energy. Not perfectly, but within ranges tight enough that enzymes can keep working and structures can keep their shape. This regulation does not happen once in a while. It happens continuously, even while you sleep tonight, even while your breathing slows and your body becomes still. Your cells remain active in their own quiet way, holding their internal world steady. So when we talk about a 24-hour period in the life of a cell, we are not describing a story with chapters the cell can feel. We are describing a pattern imposed from outside. The body creates rhythms. Light enters the eyes. The brain responds. Hormones rise and fall in the blood. Nutrients arrive in waves after meals and then fade. Body temperature drifts up and down by small amounts. A cell does not see light. What it sees are chemical consequences. Hormone binding to a receptor, a higher level of glucose, a slight change in oxygen availability, a shift in calcium concentration. From the cell's point of view, time is chemistry changing. Most human cells contain internal molecular clocks that respond to these repeating patterns. These clocks are built from genes and proteins arranged in feedback loops. A protein is produced. As it accumulates, it suppresses its own production. Over time it degrades. As it disappears, production begins again. This rise and fall takes roughly a day. Not perfectly. Not without drift. These clocks need to be nudged back into alignment by signals from the body. But they are close enough that a cell can anticipate regular changes rather than only react. Even so, it is important to understand what the clock is doing. It is not telling the cell what to do. It is biasing what the cell is already doing. Energy production continues. Repair continues. Communication continues. Protein turnover continues. Nothing stops so that the clock can run. Instead, the clock shifts emphasis, like a tide changing the direction of a current without changing the ocean. In what we call morning, many cells are bathed in signals that slightly favor energy use. Nutrients are more available. Hormones lean toward mobilizing fuel rather than storing it. Enzymes that break down molecules are a little more active. In what we call night, the balance drifts the other way. Synthesis slows slightly. Maintenance becomes a little more prominent. The cell conserves without ever becoming inactive. None of this is dramatic. There is no moment when the cell suddenly becomes busy or suddenly becomes calm. Instead, there are thousands of small adjustments, each one almost insignificant on its own, but together forming a repeating rhythm. One of the most important things to understand about a human cell is that it does not exist for today. It exists to persist. Every process inside the cell is shaped by the need to remain functional not just for hours, but for years, even decades. For some cells, an entire lifetime. This is why the cell is conservative. It avoids extremes. It favors stability over speed. It builds in redundancy. It tolerates small errors because eliminating every mistake would cost too much energy and time. When damage occurs and damage is constant, the cell does not panic. It repairs what it can, isolates what it cannot, and continues. This attitude shapes what a day looks like from inside. Rather than events, the cell experiences balance. How much energy is coming in versus going out? How much damage is accumulating versus being repaired? How much protein is being made versus recycled? Over 24 hours, these balances shift slightly, then shift back again. A day in the life of a cell is less like a story and more like a tide. Slow movement in one direction, then a slow movement back. Nothing abrupt, nothing urgent, just continuous adjustment. As we move through this series, we will follow that tide. We will begin at the cell's boundary, at the thin moving skin that separates its crowded internal sea from everything outside it, and we will see how that boundary quietly controls almost everything the cell experiences. The membrane is also where the cell listens. Scattered across the surface are receptors, proteins that recognize specific molecules in the extracellular fluid, hormones like insulin, growth factors like EGF, neurotransmitters like acetylcholine, cytokines released by immune cells. When one of these molecules binds to its receptor, the membrane does not simply let it inside. Instead, binding changes the receptor's shape. That change is transmitted to the inside of the cell, triggering reactions. A message crosses the membrane without anything physical needing to cross. Inside, enzymes are activated. Ion channels open. Calcium may be released from internal stores. Gene expression may shift over minutes to hours. All of that can begin with one binding event on the surface. Over a day, the pattern of signals arriving at the membrane changes. Hormone levels rise and fall. Nutrients arrive in pulses. The chemical environment outside the cell is never quite the same from 1 hour to the next. The membrane responds continuously. Transport rates adjust. Receptors are made more or less sensitive. Some receptors are pulled inward and recycled, a process called endocytosis, allowing the cell to quiet a signal that has become too strong. New receptors are inserted when sensitivity needs to increase. Another constant feature of the membrane is renewal. Lipids are damaged by oxidation. Proteins lose their shape. Receptors bind too much or too little. The membrane cannot be allowed to drift. So patches of membrane are continuously removed and replaced. Vesicles bud inward. Others fuse outward. The cell's surface is quietly rebuilt as it is used. The boundary is also where the cell physically connects to its world. Cells bind to other cells. They anchor themselves to surrounding scaffolds. They sense tension and pressure through membrane proteins linked to the internal skeleton. Mechanical forces become chemical signals. As the body moves, rests, and sleeps, these forces change. The membrane translates them into small adjustments in behavior. Taken together, the membrane is not merely a barrier. It is a filter, a sensor, a communicator, and a stabilizer. It defines what the cell is exposed to and what it is protected from. And across the entire day, It quietly maintains the conditions that allow everything inside the cell to continue without interruption. In the next chapter, we will move inward to the systems that keep that continuity possible at all. We will look at how cells generate usable energy and how mitochondria, with their internal membranes and gradients, keep the cell supply from moment to moment. Chapter 3. Energy inside the cell. Every action inside a cell, no matter how small, has a cost. Molecules do not move for free. Bonds do not form or break without an exchange. Order does not maintain itself without continual input. The cell survives because it has a way to manage energy steadily. Inside a human cell, energy is stored and transferred in chemical form, not as heat, not as motion, but has potential locked inside molecular bonds. The most important of these molecules is ATP, adenosine triphosphate. ATP is not energy itself. It is a carrier, a portable packet of chemical potential that can be spent anywhere in the cell. When the cell needs to build a protein, move an ion, reshape its membrane, or repair damage, ATP is what makes that possible. And yet, ATP is not stored in large reserves. Cell keeps only a small supply at any moment, enough for seconds, not minutes. So ATP must be regenerated continuously. Some ATP is made in the cytoplasm through glycolysis, where glucose is split into smaller molecules. But in most human cells, the majority is produced by mitochondria. Mitochondria are often called the power plants of the cell, but it is better to picture them as quiet engines that never stop running. Each mitochondrion is enclosed by two membranes. The outer membrane separates it from the rest of the cell. The inner membrane folds inward, forming deep ridges and surfaces. It is across this inner membrane that the main conversion happens. Nutrients arrive in partially broken down forms. Sugars, fats, and sometimes amino acids are fed into reactions that strip electrons away from them. Those electrons are passed along a chain of proteins in the inner membrane, often called the electron transport chain. As electrons move through this chain, small amounts of energy are released step by step. That released energy is used to pump protons, hydrogen ions, from one side of the inner membrane to the other. Slowly, a gradient builds. There are more protons on one side than the other, and that imbalance stores potential energy, like water held behind a gate. Helpful image here is a slow water wheel. Imagine a stream that never stops flowing. If you build a small dam, water piles up behind it. When you allow water to flow back through a narrow opening, it can turn a wheel steadily, quietly, without splashing. The proton gradient is like that. The membrane is the dam. The protons are the water. The wheel is a protein complex called ATP synthase. Protons flow back across the membrane through ATP synthase, and that flow drives the assembly of ATP. This is not a dramatic process. No sparks, no bursts, no noise, just steady conversion. Oxygen plays a crucial role because it accepts the electrons at the end of the transport chain. Without oxygen, the chain backs up and proton pumping slows, reducing ATP production. This is why oxygen availability matters so deeply to cells, even though they never experience air. Over the course of a day, the demand for ATP changes slightly. When the body is active, cells tend to use ATP faster. When the body is resting, demand eases, but energy production does not stop when demand is low. Mitochondria continue to maintain gradients. ATP continues to be made. The cell keeps supply available because waiting is dangerous. If a sudden demand arrives, a pump must run, a signal must be transmitted, a repair must happen. The cell cannot afford to pause while energy systems restart. So instead, the cell accepts some inefficiency for the sake of reliability. Keeps the water wheel turning. Mitochondria themselves change over time. They fuse and split. They are repaired, recycled, and sometimes removed if they become damaged. Over hours, Their activity drifts gently in response to signals and need. But again, nothing switches fully on or fully off. There is only adjustment. From the inside, energy is not something the cell seeks. It is something the cell maintains. Moment after moment, hour after hour, ATP is broken down and rebuilt, supporting every other process in the cell without drawing attention to itself. In the next chapter, we will turn to the molecules that consume much of this energy, the proteins that carry out most of the cell's work, and the quiet cycle of making, folding, using, and replacing them that continues without interruption. Chapter 4. Making and replacing proteins. If you could remove the water from a human cell and leave only what does work, what would remain would be mostly protein. Proteins are the cell's hands, tools, scaffolds, messengers, and machines. They catalyze reactions, move molecules, transmit signals, form structures, and regulate almost everything that happens. A cell is not defined by having proteins. It is defined by which proteins it has, how many of each, and how long they remain. Proteins are built as long chains of smaller units called amino acids. The instructions for these chains are stored in DNA, but the act of building happens elsewhere. In the cytoplasm, ribosomes move slowly along messenger RNA molecules, reading their sequence and linking amino acids together one by one. This happens continuously. Even in a cell that appears quiet, ribosomes are at work. New proteins are being made to replace old ones, damaged ones, or simply those no longer needed. When a protein chain is finished, it is not yet useful. It must fold. Each chain collapses into a precise three-dimensional shape, guided by the chemical properties of its amino acids. Many proteins fold on their own. Others need help. That help comes from molecular chaperones, proteins whose role is to assist other proteins in folding correctly. They do not dictate the final shape. They prevent mistakes while the protein finds it. Only when folded correctly can a protein do its job. Some proteins last minutes, others last hours. A few persist for days. None are permanent. Heat, oxidation, and mechanical stress slowly damage proteins. Random chemical interactions distort their shape. Even without damage, many proteins simply wear out through use. The cell does not try to preserve proteins indefinitely. It relies on turnover. When a protein becomes damaged or unnecessary, it is tagged with a small molecule called ubiquitin. This tag marks it for removal. The protein is delivered to a structure called the proteasome, a molecular recycler that pulls proteins apart into amino acids. Those amino acids are reused. Nothing is wasted. This constant cycle of synthesis, folding, inspection, and degradation consumes a large fraction of the cell's energy, and yet it is one of the reasons cells are so resilient. Rather than relying on static parts that must never fail, the cell relies on renewal. If a protein is slightly flawed, it does not cause catastrophe. It is replaced. If conditions change, the cell does not redesign itself. It shifts which proteins it makes more of, and which it allows to fade away. Over the course of a day, these patterns shift gently. At some times, proteins involved in metabolism are produced a little more often. At others, proteins related to repair, stress tolerance, or recycling become slightly more prominent. There is no moment when protein production stops. There is no moment when replacement suddenly accelerates. There is only flow. The internal scaffolding of the cell, which appears solid, is also in motion. Filaments assemble and disassemble while the overall shape remains stable. This is one of the quiet paradoxes of cellular life. Stability is achieved not through permanence, but through continuous change. From 1 hour to the next, the cell is not made of exactly the same proteins, and yet it behaves the same way. It remains recognizably itself. This ongoing renewal is how reliability is maintained in an environment where damage is unavoidable. In the next chapter, we will look outward again, at how cells exchange information with one another, and how trillions of independent units coordinate their behavior without any central control. Chapter 5. Communication and Signaling. A human cell is never alone. Even when surrounded by still tissue, it exists in a sea of chemical messages. Signals arrive from nearby cells, from distant organs, and from the cell's own internal compartments. There is no silence. What is striking is that most of these signals are not commands. They are influences. Cells do not operate under orders. They operate under bias. The molecules that carry information are often small and simple. Hormones like insulin, neurotransmitters like acetylcholine, growth factors, cytokines, gases such as nitric oxide. Each signal drifts through fluid, encounters the cell's surface, and may bind to a receptor embedded in the membrane. When that happens, nothing dramatic occurs. There is no jolt. The receptor protein changes shape slightly. That shift alters how it interacts with proteins inside the cell. From there, a cascade begins. Proteins activate other proteins. Enzymes add phosphate groups. Second messengers, such as cyclic AMP or calcium, briefly rise in concentration, then fall again. One molecule outside can influence thousands inside. This amplification gives signals reach, not force. A classic example is adrenaline. When it binds to a beta-adrenergic receptor, it activates a G protein, which increases cyclic AMP, which activates protein kinase A. That cascade alters enzyme activity across the cell, making energy more readily available. The same signal in a different cell can produce a different effect. Acetylcholine causes muscle cells to contract, but slows the heartbeat when it binds to receptors in heart cells. The signal is the same. The response depends on the receptors present and the internal state of the cell. This is why signals bias rather than command. They make certain reactions more likely. Inside the cell, signaling pathways overlap and intersect. Calcium signals influence secretion and contraction. MAP kinase pathways affect gene expression. PI3K and ACT pathways adjust metabolism and survival. These pathways form networks, not lines. Noise is unavoidable. Molecules collide randomly. Signals overlap. Concentrations fluctuate. Cells are designed to tolerate this. Feedback loops dampen extremes. Receptors become less sensitive when signals are strong, a process called desensitization. Some receptors are pulled into the cell and recycled, preventing overreaction. Over a day, the pattern of signals changes slowly. Hormones rise and fall. Nutrients arrive in waves. Mechanical forces shift as the body moves and rests. The cell integrates all of this continuously. Even the cell's internal clock is part of this conversation. Clock genes produce proteins that suppress their own production, degrade, and allow the cycle to begin again. The clock does not dictate behavior. It nudges it. It biases the cell towards certain responses at certain times, without overriding other signals. Communication at the cellular level is not about messages being delivered. It is about probabilities being adjusted. Moment by moment, the cell listens, responds, and rebalances, never obeying, never ignoring, always integrating. Chapter 6. Repair and Maintenance. If you could watch the molecules inside a cell closely enough, you would notice something unsettling. They are constantly being damaged. Chemical bonds snap under heat. Reactive oxygen species alter proteins and DNA. Water attacks vulnerable links. Mechanical stress distorts structures. This is not failure. It is the normal condition of life at the molecular scale. What keeps a cell alive is not the absence of damage, but the presence of repair. DNA, often imagined as stable and permanent, is inherently fragile. Bases are lost spontaneously. Cytosine is converted to uracil. Radiation introduces breaks and distortions. Left alone, DNA would decay. The fact that it does not is because cells repair it continuously. Thousands of DNA lesions occur in a single cell every day. Most are corrected almost as soon as they appear. Enzymes patrol the genome, scanning for irregularities. When damage is found, a small section is cut out, resynthesized and sealed. Different repair systems handle different problems. Some fix single damaged bases. Others remove bulky distortions caused by ultraviolet light. Others correct copying errors. These systems operate all the time. They do not wait for damage to accumulate. Proteins face similar challenges. They misfold. They oxidize. They lose function. Molecular chaperones attempt to refold them. If that fails, ubiquitin tags them for removal. The proteasome dismantles them into reusable pieces. This ongoing management of protein quality is called proteostasis, and it consumes large amounts of energy. Membranes are renewed as well. Lipids oxidize. Embedded proteins drift out of optimal shape. Vesicles remove damaged patches and deliver replacements. Even organelles are maintained. Mitochondria that become inefficient are removed through mitophagy. New ones form through fusion and division. Over the course of a day, much of the cell's interior is quietly rebuilt, and yet the cell looks the same. This is the core principle of cellular maintenance, stability through renewal. The cell does not aim to preserve individual molecules. It aims to preserve function. Small problems are handled immediately, preventing larger ones from emerging. Damage is constant. Repair is constant. Failure is rare because maintenance never stops. From the cell's perspective, there is no special time for fixing things. Repair is woven into every moment. As the day continues and signals shift, maintenance remains in the background, ensuring that when the next demand arrives, the cell is still intact, still balanced, and still able to continue. In the next chapter, we will move to the center of the cell, to the nucleus, and look at how access to genetic information is regulated, allowing the cell to remain itself while still responding to change. Chapter 7. The Nucleus and Gene Control. At the center of most human cells sits the nucleus. It is enclosed by a double membrane, punctured by thousands of tiny openings called nuclear pores. Through these openings, molecules pass constantly. RNA leaves. Proteins enter. Signals move back and forth. Nothing is sealed shut. Despite this activity, the nucleus is often misunderstood. It is not a command center issuing instructions in real time. It is not a book that is read from beginning to end each day. It is closer to a dim archive. Inside are shelves of information, tightly packed and carefully guarded. Most of it remains closed. Only selected sections are opened, copied, and used. Every human cell contains essentially the same DNA, tens of thousands of genes. And yet, a liver cell behaves nothing like a neuron, and a muscle cell behaves nothing like a skin cell. The difference is not the information stored, it is which information is accessed. Most genes are silent most of the time. Their DNA is wrapped tightly around proteins called histones, forming a compact structure known as chromatin. In this state, the genetic code is physically inaccessible. When a gene needs to be used, the chromatin around it loosens. Chemical tags placed on histones act like bookmarks, marking regions as open or closed. Acetylation tends to open access. Certain methylation patterns tend to close it. These changes do not alter the DNA sequence. They alter access. This layer of control is sometimes called epigenetic regulation, and it allows cells to remain flexible without losing identity. Another layer of control comes from transcription factors. These are proteins that bind specific DNA sequences near genes and influence whether transcription begins. They respond to signals from the cytoplasm, which means events at the membrane can influence gene activity without ever touching DNA directly. If insulin signaling is strong, transcription factors promoting metabolism enter the nucleus. If stress signals dominate, factors involved in repair and protection take precedence. Even after a gene is transcribed, control continues. RNA molecules are edited through a process called splicing, where sections are removed or rearranged. A single gene can produce multiple proteins depending on how its RNA is processed. The nucleus, then, does not dictate behavior. It permits certain behaviors and restricts others. Over the course of a day, gene expression patterns drift slightly. Clock genes such as TLOCK and BNAL1 produce proteins that suppress their own production. As these proteins accumulate, they close access. As they degrade, access opens again. This cycle takes roughly 24 hours. The effect is modest. Only a small fraction of genes show strong daily rhythms, and even then the changes are usually subtle. A gene may be slightly more active in the morning and slightly less active at night. Rarely does it switch fully on or fully off. Different tissues show different patterns. Liver cells adjust genes involved in metabolism and detoxification. Muscle cells adjust genes related to contraction and energy use. Brain cells change very little, reflecting the need for stability. If a strong signal arrives, timing is overridden. Damage activates repair genes regardless of the hour. Nutrient influx activates metabolic genes even at night. Time is one influence among many. Across the day, the nucleus quietly manages access to information, ensuring that the cell remains what it is, while still adapting to the chemistry of the moment. Chapter 8. Adaptation across the day. A human cell exists inside a body that is always changing. Light levels rise and fall. Body temperature drifts. Hormone concentrations oscillate. Meals arrive, then stop. Muscles contract, then relax. The cell must adapt to all of this. But adaptation does not mean reacting sharply to every change. It means smoothing fluctuations so that internal balance is preserved. One of the clearest examples of this is metabolism. Metabolic pathways are not rigid pipelines. They are flexible networks of reactions whose relative activity can shift. When nutrients are plentiful, pathways that store energy become slightly more active. When nutrients are scarce, pathways that release stored energy are favored. These shifts are guided by molecules that reflect energy status. ATP signals abundance. AMP signals scarcity. AD+ reflects the balance between oxidation and reduction. Enzymes such as AMPK respond to rising AMP by favoring energy production and conservation. Pathways associated with growth and synthesis ease back. When nutrients are abundant, signaling through pathways like mTOR biases the cell toward building and expansion. Circadian signals add another layer. In liver cells, glucose production, lipid synthesis, cholesterol metabolism, and detoxification peak at different times of day. This separation reduces competition for resources and prevents conflicting reactions from running simultaneously. Adaptation also occurs at the level of organelles. Mitochondria adjust their shape and connectivity through fusion and division. During periods of high demand, mitochondrial networks become more interconnected. During lower demand, maintenance and quality control become more prominent. Autophagy plays a role here as well. Damaged proteins and organelles are enclosed in membranes and delivered to lysosomes for breakdown. The resulting components are reused, providing materials when external input is low. Importantly, adaptation is limited. Cells cannot reconfigure themselves completely every few hours. Too much flexibility would be destabilizing. So adaptation is layered on top of stability. DNA repair continues regardless of time. Harn gradients are maintained. Protein turnover does not stop. If a meal arrives at an unexpected hour, the cell responds. If activity occurs at night, pathways adjust, but responses are measured. The cell does not chase every fluctuation. Over a day, adaptation looks less like reaction and more like drift. Small changes in enzyme activity, slight shifts in transporter abundance, gradual redistribution of resources. From the outside, the body seems to move through distinct phases. From inside the cell, those phases blur into one another. Adaptation is continuous negotiation, carried out molecule by molecule, without pause. Chapter 9. Moving toward rest. As the external world shifts toward evening, the chemical environment surrounding each cell begins to change. Light exposure decreases, hormone levels adjust, body temperature drifts downward by fractions of a degree. Nutrient input becomes less frequent. From the cell's perspective, none of this arrives as a signal labeled rest. It arrives as a gentle change in balance. Cortisol levels fall. Insulin pulses become less frequent. Melatonin begins to rise. These molecules interact with receptors on the membrane and bias internal pathways. They do not tell the cell to stop. They change which processes are favored. Energy production continues. Mitochondria maintain their gradients. ATP is still generated, but the demand profile shifts. Processes associated with rapid growth and expansion ease slightly. Processes associated with maintenance and quality control gain weight. Protein synthesis continues, but the mix changes. Ribosomes translate more transcripts related to stress response and repair. Fewer resources are directed toward proliferation. Energy sensing pathways respond to changing conditions. Rising AMP and NAHI+ activate enzymes that favor conservation. Anabolic pathways slow. Catabolic and recycling pathways become more prominent. Autophagy increases gradually. This is not starvation. It is housekeeping. Worn proteins, damaged membranes, and inefficient organelles are enclosed and delivered to lysosomes. Their components are recycled. Nothing dramatic happens. The cell does not shut down. It shifts emphasis. Even the membrane reflects this change. Receptor sensitivity adjusts. Some receptors are internalized. Others become more responsive. The cell becomes slightly less reactive to signals demanding immediate action, and slightly more receptive to signals associated with maintenance. This transition is slow. There is no clear boundary between active and resting states. There is only a gradual rebalancing, guided by overlapping signals and constrained by the need for stability. As you settle into rest, your cells do not sleep. They prepare. They conserve. They continue. In the next chapter, we will follow this shift further into what we call night and see how cells use this period to focus on deeper maintenance, repair, and quiet recalibration. Chapter 10. Nighttime activity. Night, from a cellular point of view, is not a pause. It is a shift in emphasis. As external signals associated with darkness and rest continue, the internal environment of the cell settles into a different balance. Melatonin levels are higher. Core body temperature is slightly lower. Feeding-related hormones are quiet. None of this tells the cell to stop. It tells the cell that competing demands are fewer. This creates space. One of the main activities that becomes more prominent during this period is quality control. Proteins that have accumulated minor damage during the day are more likely to be inspected. Molecular chaperones spend more time assisting misfolded proteins, either guiding them back toward functional shapes or handing them over for removal. In the endoplasmic reticulum, where many proteins are folded and modified, sensors monitor the balance between incoming proteins and folding capacity. When misfolded proteins accumulate, a response is triggered that temporarily slows new synthesis and increases repair and degradation. This is not an emergency response. It is routine housekeeping. Autophagy often becomes more active at night. Sections of cytoplasm are enclosed within membranes, forming vesicles that deliver their contents to lysosomes. Inside, enzymes dismantle proteins, lipids, and organelles into reusable components. Mitochondria receive particular attention. Those that have become inefficient or damaged are selectively removed through mitophagy. Others fuse together, sharing contents and evening out damage. Over time, this improves the overall efficiency of the mitochondrial population. DNA repair continues as it always does, but the cell may devote slightly more resources to correcting damage accumulated during periods of higher metabolic activity. Oxidative lesions are resolved, breaks are sealed. At the same time, the molecular clock resets. Proteins produced earlier in the cycle, such as PER and CRI, are gradually degraded. As they disappear, repression of their own genes lifts, allowing a new cycle of transcription to begin. This resetting does not interrupt other processes. It unfolds quietly alongside them. Night is also a time of preparation. Metabolic intermediates are adjusted. Energy stores are stabilized. Transporters are repositioned. The cell subtly reshapes itself so that it can respond smoothly when external signals shift again. From the outside, night appears still. From inside the cell, it is a period of careful inspection, recycling, and recalibration. Nothing stops. The work simply becomes less visible. Chapter 11. Error Tolerance. At the scale of molecules, mistakes are unavoidable. Enzymes occasionally bind the wrong substrate. Ribosomes insert the wrong amino acid. DNA polymerases make rare copying errors. Proteins fold imperfectly. Signals overlap or arrive at awkward moments. If cells demanded perfection, they would fail almost immediately. Instead, they are built to tolerate error. Consider an enzyme designed to bind glucose. In a crowded cytoplasm, it may occasionally bind a similar molecule, such as galactose. The fit is imperfect. The interaction is weak. The molecule drifts away, and the enzyme tries again. No harm is done. This pattern repeats across countless reactions. DNA replication provides another example. DNA polymerases are highly accurate, but not flawless. Proofreading enzymes correct most mistakes as they occur. Mismatch repair systems scan newly copied DNA and fix errors that slip through. Even so, a small number of changes remain. Most are harmless. Some are corrected later. A very few persist. The cell does not collapse because of this. Redundancy limits the impact. Many genes exist in multiple copies. Many pathways have alternative routes. If one component falters slightly, others compensate. Protein quality control adds another layer. Misfolded proteins are recognized and handled before they can interfere with other processes. Chaperones attempt refolding. Degradation systems remove persistent problems. Signaling pathways are also designed to be tolerant. Receptors bind ligands briefly. Cascades integrate signals over time rather than responding to single events. Feedback loops dampen spikes and prevent runaway activation. This tolerance is essential. If every fluctuation triggered a full response, the cell would be unstable. If every error caused shutdown, the system would grind to a halt. Instead, the cell treats error as expected. It corrects what matters. It ignores what does not. Over a single day, countless small mistakes occur and disappear without consequence. A protein misfolds and is replaced. A DNA base is damaged and repaired. A signal is misread and overridden by others. The cell survives not by eliminating error, but by containing it. Error tolerance is not a flaw. It is a design principle that allows life to persist in an imperfect environment. Chapter 12. Continuity. When we describe a day in the life of a cell, we draw lines that the cell itself does not recognize. There is no beginning. There is no end. From the cell's point of view, there is only continuity. Energy production flows into energy use. Protein synthesis blends into degradation. Damage leads into repair. Signals rise and fall. Gene activity drifts and returns. Morning, afternoon, evening, and night are gradients, not boundaries. When one day ends and another begins, nothing resets. The ATP being used now was made moments ago by mitochondria that have been active for years. The proteins carrying out reactions were built hours or days earlier. The DNA being repaired tonight may have been damaged yesterday. The cell carries its past forward, and yet it is not burdened by it. Because renewal is constant, history does not accumulate uncontrollably. Proteins are replaced. Lipids are recycled. RNA is degraded and resynthesized. Organelles are renewed. The cell remains recognizably itself, even though its components change. This is the quiet paradox of cellular life. Nothing is permanent, yet identity persists. Across days, weeks, and years, the same patterns repeat. Boundaries are maintained. Energy is managed. Information is regulated. Damage is repaired. Some cells persist for an entire lifetime. Others divide, passing their internal balance to their descendants before disappearing. In both cases, continuity remains. What changes slowly is not the daily rhythm, but the capacity to maintain it. With time, repair becomes less efficient. Errors accumulate. Renewal slows. But for most of a cell's existence, this failure is distant. Day after day, the cell continues, not because it follows a schedule, but because it never stops adjusting. If you could observe a single human cell continuously, you would not see events. You would see flow, balance maintained through change. And as this day ends and another begins, the cell remains already partway into what comes next, carrying forward the chemistry that allows it to persist moment by moment without pause. And so it continues,